And hi there, Molec and Cell friends. Good afternoon, good morning. This is Sunday afternoon. There you go. Hey, we're getting set to do the first lab. And what we're going to do is practice using micropipettes. And additionally, we're going to build a standard curve for determining protein concentration, and hopefully by the end of the semester, when we do some animal parts, we'll be extracting proteins and there'll be an unknown concentration, and we can use this standard curve to determine the protein concentration. So let's see. First, allow me to introduce the micropipettes. Say hi. This is what's known as a P1000. This is a P200, and this is a P20. So we got the mama bear, the baby bear, and then I don't know. All right, regardless.
Anemic infant bear.
So these are micropipettes, and they can be a little confusing at first to use. There, you see the numbers? Anyway, let me show you how, just a brief rundown of how they work. They all have 4 window little numbers on them that you can dial around and dial and change. And the way they work is you have a lock here on this little plunger, pull that up, and then you can begin dialing down and changing the number down. So, all right. It takes a little bit of experience and practice to get good with these, but that's what this is about. So, the great big one with the blue tip or cap on it, called the P1000, it uses these blue tips. And just observing here, you touch down just very gently, give it a little tap, and then give it a quarter twist. All right? So we'll go through dispensing in just a moment, but let me show you how they're calibrated and how they work. So we got these different sets of pipetters, and the P1000 blue tip, it's designed to go from 1,000 microliters, i.e. 1 mil, down to, about 200 microliters. And it's got, when you're looking at the squares, it has 4 squares, 4 numbers on it, and 1, 0, 0, 0. That is 1 mil. So if you wanted to pipette 300 microliters, you'd dial this down to where it read 0, 3, 0, 0. So that would be 300 microliters. Okay, so that's the P1000. We don't use the P1000 for too much, mainly because we don't use big volumes in molecular genetics and molecular cell. This week's lab, we actually will be doing 1 mil shots of some stuff, and so yeah, you could use it a bit. All right, the next one I use a lot, is the P200. The P200, this is a nice one in that it has the yellow button on it. It has a tapered, a much skinnier barrel, but comparing it to the P20, you'll see the P20 has a much skinnier barrel. Yeah, there you go. You can see that. So that's the P20. All right, so the P200, it's using yellow tips, and the same thing. You touch it gently. You don't have to jab, and you don't want to jab too hard here. Just a bit of a tap. And then betwixt thumb and forefinger, give it a quarter turn crank. You're ready to rock. All right. This P200 has also 4 numbers on it. You can see that. There's a dash at the bottom, which is essentially a decimal. And so this is in microliters. And so here's what the different squares would indicate. let's see how this contrast-wise, is that good? Can you see that? Yeah, not bad. All right, so you got the 4 numbers now here, and this one has the line across it. So the P200 is designed to run from volumes of 200 microliters down to 20 microliters. Although these P200s actually are pretty good, and they are easily good down to 10 microliters. Below 10, they're not as great. All right, so 2000, that's 200 microliters. Just by the way, if you set a P1000 up to that, you'd break it because they don't go up to 2 mils. These go up to 200. All right, so the squares look the same between the two, but the numbers actually have different meanings and different volumes. So let's say we want to pipette out 150 microliters. That would look like this, 150. Okay, so that's the P200. Can you see that? Sure, enough. All right, then finally, we have the P20. So the P20 has the real narrow tip on it. And it also has 3 numbers, or 4 squares on it. This time, though, the line is in the middle, too. And it works like this. So the P20 1, 2, 3, 4, and the line is here. The line is essentially a decimal point. And so the P20 has a volume working range from 20 microliters down to 1 microliter. You can dial down below that, but sorry, with a P20, if you're trying to pipette less than a microliter, you're kind of imagining things. They will do that, but the tip size and the surface tension of fluids make it so below 1 microliter, don't try it. A microliter is a little nub. I mean, you can accurately pipette a microliter, but below that, probably not so much. So you might have this one set for 2000. That would be 20 microliters, 20.00. And if you wanted to pipette 5 microliters, just an example, that would be 050. Okay? So what we're going to do today is a whole range of pipettings and pipette volumes. Yeah, it's a 5. And that's how the exercise is going to work. All right, so let me just give you a brief description about how you would accurately pipette up a volume. So all the pipetters, they work pretty much the same way. You dial in your number, then you go ahead and click the button down to push it all the way down. That locks the volume in so you can't accidentally change it, and that's good. I'm going to dial up to 1 mil, and boop, 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 there, 1 mil. All right, you pick up a pipetter, put your thumb on the plunger, and push down. You'll notice that when you push down, there's a first spot of where it has a stop. It's not a hard stop. If you push down further, it's spring-loaded. It's kind of the blowout. So when you get ready to pipette something, what you do is you push down to the first stop, and it's always better manners to push down to the first stop before you touch the solution, because we don't like to blow bubbles. So putting the tip into the solution and then pushing down, you're blowing bubbles, not so good. You push down, and then you'll let up slowly. Don't pop the clutch, let up slowly. Then to dispense, you push down to the first stop, and if you have anything remaining, you push out. It's kind of like the blowout. So, as an example.
Daddy, pump me like you know I like it.
Okay, I have a cheerleading section. Here is 1 mil of coffee. There's a little bit of blowout in there. There you go. All right, so an important thing is never let the fluid touch the barrel, you'd only want to touch the fluid up to the tip of your micropipette. So, just as an example here, can you see? I'm pouring my coffee out. Hold on, let's see. There we go. All right. Now, when you get ready to dispense, let me grab a test tube here real fast. Hold on a second. I'll bring
I
right
got
back.
two right here.
Hey, a glass test tube.
Oh, you need one? Big one. Just 1 second.
Huh?
Just 1 second, guys. We'll edit this out in post.
He's gonna be more. All right. So now, when you get ready to administer, these are little small tubes. We'll be using tubes a little bit bigger than this. What you want to do is pipette up, and it's not the best to add it sort of as a hanging drop. It's better. Don't want to waste coffee. To always touch either the existing solution, that you have in the bottom of the tube, or touch the side of the glass tube, like so, and dribble down. Now, what you don't want to do is have your samples way up high, so they're not down at the bottom, because today's operation is going to be using a spectrophotometer in which we need to have all the solutions down at the bottom. So it's a good idea to go ahead and bring things down to the bottom. Okay? Now, all right, so that's pipetting. It's going to take you some practice to get good at it. It takes a little bit of time and a little bit of looking at it going, is this right? Because what you don't want to do is pipette in 200 microliters when you're meant to pipette in 20 microliters. So you have to be a little bit careful with it. Today what we're going to do is use these pipetters to set up a series of protein concentrations, and we're going to do a protein assay. And so, just a little background about protein assays. Why do you need to do protein assays? You want to know how much of a protein you have in solution. Like if you're doing enzyme kinetics, you need to know how much enzyme you got. If you're trying to purify stuff, you want to know how much you have. Knowing how much protein is important. Like what we're going to be doing is comparing different extraction techniques and seeing what produces or what pulls out the most protein. So protein assays are a useful thing. There are a number of ways you can assay proteins. You can use UV spectrophotometry. In that, proteins will absorb UV light, particularly the tyrosine ring. Tyrosine, our group, absorbs UV light really well. UV light is not particularly accurate for proteins because it depends on the amino acid composition. And so generally, we use UV detection as a way of saying, yeah, there's protein in this sample, but not for determining how much protein is in the sample. Additionally, other biomolecules can be UV absorbent too. DNA is. So it's a good detection, general detection tool, but it's not a particularly quantitative detection tool. For quantitative detection, what is typically done is some kind of interacting chemical or a dye. And there are a number of different protein assay reagents. A standard one is known as the Bradford reagent. This is using a blue dye. Just for all you nerds in the audience, is something known as Kumasi Blue G250. Not that anybody cares, but it's actually a dye that's used in the clothing industry. And it works fairly well. Kumasi Blue G250, we use Kumasi Blue R250 as a standard protein dye when we're looking at proteins in electrophoresis in their gels. But for dye binding, it works fairly well. The problem is there are a lot of things that will interfere with this, that'll inhibit the Bradford reagent from binding, or that'll give you a false positive and increase the dye binding. It's just not as accurate either. It's not as sensitive as we might need. What we're using, to be perfectly honest, is a reasonably high-tech but really nice protein assay. You find yourself in a research lab, you very likely will be using something called excuse me, the bicanoic acid protein reagent, which is abbreviated as the BCA reagent. BCA is a proprietary material from the Pierce Biochemical Company out of Milwaukee, Wisconsin. And it is a lovely sort of apple green. But then when you add proteins to it, it begins to turn purple. And we can measure that color change in the spectrophotometer. So, what we're going to do is set up a series of protein concentrations and develop what we call a standard curve. Let me show you how. So, the relationship between the absorbance and dye binding and protein concentration is quite linear, which is nice, because that allows us to develop a graph that'll look something like this. A standard way of doing this would be to have the the spectrophotometer absorbance. And spectrophotometer absorbance is a unitless measurement. It's called absorbance units full scale. So there's not a units for absorbance, it is just absorbance. Or it's abbreviated sometimes in the biz as absorbance units full scale, or AUFS, absorbance number. And down on the x-axis, we're going to have the protein concentration. And we'll have a blank, a 0, and then we will develop a standard curve that, if you're good, will be a really good linear equation. And so you can use a statistical calculator or Excel, we can talk about how you can do it, and you can get the linear equation. And if you're good, the correlation coefficient, that is how close your points lie on this theoretical line, will be really very, very high. How high? I don't know. Let's see. We'll have a little competition and see who can do the best. It's not uncommon to have a correlation coefficient of 0.95, 0.97. You know, it's not that hard to do. And all right, so from here you can get the linear equation that is y equals mx plus b, where we've got the y value is equivalent to the slope times the x plus the y-intercept. And basic algebra, if you don't get this, hey, talk amongst yourselves, go to Excel, whatever. We know about it. All right, so let me show you what we're going to do then. The protein concentration range that we're going to use is kind of designed to give us a nice line and give you some experience pipetting a range of different volumes. So in order to do the BCA assay, it's really pretty simple. You take 0.1 mil of your sample and 2 mils of the BCA reagent, and mix it up 37 degrees for 30 minutes, but truthfully we usually go about 15 minutes and it's just fine. And you then read these on the spec. I think 542 nanometers. We got some very nice spectrophotometers here, and they're really easy to use. Read the instructions on it that's written in the handout, and we'll go through it real fast as well. Basically, you put your blank in, hit set blank, put your tube in, close the lid, wait a moment, read the number, move on. All right. So what we're going to do then is set up a series of concentrations like so. We're going to have 0 protein in the sample. That's easy. That's just going to be straight diluent solution. We're going to have 50, 75, 100, 200, 300, 500, 1,000 micrograms of protein per mil. So this is micrograms per mil. So in order to do these, we actually have to have what we think of as sort of a standard protein solution. And what we're going to use is a 1 mg per mil solution of BSA, as it's known in the business. This is bovine serum albumin. Albumins are a general class of very soluble proteins. Serum albumins are a very soluble protein found in blood. Bovine means cow. What you do is you take some cow blood and you pull out the very soluble serum albumin protein. You can precipitate it with cold ethanol really effectively, and you can make a near pure prep of this stuff. And we got a bottle of it, dehydrated, little crystals of protein. Tap them out, resuspend them. That is the BSA protein standard. 1 mg per mil. 1 mg per mil, bless the metric system, is the same as 1,000 micrograms per microliter. So this is in micrograms. All you got to do is load that much in microliters into 1 mil. Okay. Now here is a little bit of a cheat. The diluent that we use, that is the stuff we're going to be adding to dilute the sample, is going to be today, it could be water, but we typically use phosphate buffered physiological saline. What that means is it's got a pH of 7.2 and a salt concentration of 0.9%. This is just a standard sort of solution for molecular cell work. Phosphate buffered saline, also known, of course, in the biz as PBS. We go through liters of this when we're doing different kinds of experiments. Like immunology, you often use PBS a lot. So we're going to be using PBS. Now, if we go through this chart, we can figure out how much we're going to add. And here is where we're going to have a little bit of a potential cheat that makes things a little bit easier. If we wanted to make 1 mil of solution, we would add 50 microliters of BSA and then 950 microliters of PBS. That would be the equivalent of 50 micrograms per mil. And then, because with the BCA assay, you use 100 microliters, you take 100 microliters out of this and use it. All right, this is a bit confusing, but it is a way of saving steps and saving glassware and plastic and making things go a little bit faster. So think about it, get it in your head, but if you don't get it, just do what I'm telling you and you'll be square. Okay, you'll be fine. Rather than doing a mil, let's make up the same concentration But now let's do it in 100 microliters, or 0.1 mil. And that means that we don't have to do any second pipetting. We don't have to have a second tube. We can simply add 2 mils of BCA to these tubes. Trust me, you'll love it. It's a lot easier. Here's how you do it. You are simply moving the decimal over. So now it'll be 5 microliters of BSA and 95 microliters of phosphate buffered saline. With this one, it's 7.5 to 92.5. Here it's 10 to 90, 20 to 80. So you can use a P20 for these first set, and now you have to shift over to a P200 for these last three, 30 microliters and 70, 50 microliters and 50 microliters of PBS, and then finally 100 microliters of straight BSA. So you'll put this into a test tube, designated test tube. You're going to label up here because the sample will be down here. You don't want to have labeled out here because then it'll be interfering with the spectrophotometer. You then add to each one 2 mils of the BCA reagent. We've got these delightful vortex mixers, which I'll show you how we use them when we're in lab. You vortex it up, 30 minutes at 37 degrees, or 15 minutes at 37 degrees, whatever, and read the spec. All right, so when you come in on lab day, this is brand new to me. I'm not sure how much time we'll have. I may gas a little bit, but I won't gas on too much. You'll have everything set up. We'll get pipetting and get BCAing. All right, folks, read the handout, and I'll check in with you. We'll see you. Ciao.